Peptides are playing a rapidly growing role in therapeutics, diagnostics, and research. But here's something many teams overlook. Not all peptide testing laboratories are built the same. Choosing the wrong analytical partner can delay development, introduce regulatory risk, and slow commercialization. Today, we'll explore the top 5 things to look for in a peptide testing laboratory and how to make decisions rooted in science, not assumptions. Before we jump into the 5 key factors, let's set the stage. Peptide testing isn't just about confirming purity. It's about validating identity, understanding stability, characterizing impurities, and supporting regulatory confidence. Whether you're in biotech, pharma, CDMO environments, or academic research, the right peptide analytical approach directly impacts development speed, batch consistency, and regulatory success. The key takeaway is this: the right analytical partner accelerates timelines and de-risks commercialization. The first factor is analytical capability. Peptides are notoriously challenging to characterize. They degrade, rearrange, oxidize, and form sequence-related impurities. A serious peptide laboratory requires more than basic HPLC. Look for LC-MS/MS for identity and sequencing, HRMS for exact mass and structural confirmation, UPLC for purity and microimpurity analysis, and GC or ion chromatography for residual solvents and counterions. If a lab can only measure impurities but not identify them, you lose valuable insight for formulation work and regulatory documentation. The second factor is peptide-specific expertise. Peptides sit in the middle ground between small molecules and large biologics. They're more complex than small molecules, but not as structurally defined as full proteins. Experienced peptide labs understand challenges such as deamidation, oxidation, sequence truncation, racemization, aggregation, and salt form conversion. These issues often drive stability failures or lot-to-lot variability. Always ask how many peptide projects a lab has supported and at what level: research grade, cosmetic grade, therapeutic grade, GLP, or GMP. The third factor is regulatory alignment and documentation. For peptides entering clinical or commercial pathways, documentation isn't optional. It's a compliance requirement. Look for labs familiar with ICH Q6b for biopharmaceuticals, ICH M7 for impurity control, and GLP or GMP environments when required. A strong laboratory can generate certificates of analysis and supporting data packages that facilitate regulatory filings rather than burden them. Accurate documentation provides traceability, consistency, and evidence of compliance. The fourth factor is impurity and stability insight. Purity alone doesn't tell you how a peptide will behave over time. Comprehensive analytical testing supports synthesis improvement, lyophilization strategies, storage conditions, and even transportation logistics. Stability studies assess thermal stress, oxidation, pH sensitivity, and light degradation. Forced degradation maps vulnerable sites and validates stability indicating methods. Salt and counter-ion analysis identifies acetate, TFA, or chloride species that affect performance. And residual solvent or metal testing ensures safety and compliance. Understanding impurity lineage strengthens the entire development process. The fifth factor is scientific support. Peptide analytics is not just transactional, it's collaborative. A strong lab helps interpret complex spectra, troubleshoot synthetic routes, recommend alternative analytical methods, and support regulatory reviews. Turnaround time also matters. Academic timelines and biotech timelines are rarely the same. Ask how a lab handles urgent batches, iterative R&D, or parallel development cycles. In peptide science, data is only valuable if it's understood and actionable. Here are questions that separate commodity testing labs from strategic analytical partners. Can they detect trace impurities below 0.1%? Do they offer HRMS sequencing for peptide confirmation? Do they test counterions such as acetate, TFA, or chloride? Do they support formulation stability or only raw materials? And can they perform batch release testing and certificate of analysis creation? Peptides rarely fail because of identity. They fail due to counterions, salt forms, metal trace contamination, and oxidation. These details matter. To illustrate, here's a real-world scenario. A client submitted a peptide with 98% purity, but batches repeatedly failed stability at room temperature. Their previous lab only reported chromatographic purity. With proper LC-HRMS analysis, the root cause was revealed. Methionine oxidation combined with a counter-ion mismatch from TFA to acetate. After reformulation, shelf life tripled without changing purity. This example highlights the difference between basic testing and true analytical understanding. To conclude, the right peptide analytical partner saves time, cost, and regulatory friction. Look for core analytical capabilities, domain experience, regulatory alignment, impurity and stability insight, and collaborative scientific support. If you're developing research, therapeutic, or commercial peptides, partnering with experts can accelerate development and reduce risk. Share this presentation with your team and feel free to reach out for peptide analytical support or scientific consultation.